So here we're looking further at the muscle, kind of going to the, kind of breaking it down even further. So if you look here, so this looks similar to that bigger picture that I showed earlier. And again, we kind of tease out these different areas of the muscle. So now we're going all the way down to the myofibril, and we're looking at the contractile portion of the muscle with the actin and myosin filaments. So we're breaking down the myofibril further into a myofilament. And within the filaments, you have two different types of filaments. You have the thin actin and you have the thick myosin filaments. And these filaments are what give the muscle its striated appearance. These are also the structures that move, if you will, during what we call the sliding filament theory, which we're going to talk about in a little bit. So when our muscles shorten to create contraction, these filaments slide over one another. So you see this broken down even a little bit further. So here's your sarcomere. So the sarcomere is the smallest contractile unit of the muscle. So you can see how far the muscle's kind of getting down to this smaller and smaller level here with the actin and myosin filaments. So they're showing you the actin and myosin and what they look like. And what essentially happens is when muscle contraction happens, the actin actually slides over the myosin. So when you look at these, they're showing you here the cross bridge formation. The actin filaments actually slide over the myosin. Now, there's a couple areas of interest to note. There's what they call you'll hear sometimes them call the A band and the I band. So the A band contains the myosin, and that's the darker structure. The I band is the lighter one, that contains the actin. There's the structure called the Z line, which you can see on here as well. This is the Z line right here. And then you have what's called the H zone. The H zone is basically going to be in the center of the sarcomere. So you can kind of see that over here. Here's your H zone in here when we're looking at this individual sarcomere. So what essentially happens is the actin slides over the myosin, which causes the size of the H zone to decrease as the actin slides over the myosin. The Z lines get pulled towards the center. Okay, so the Z lines that you see right here get pulled into the center. So the process of the sliding filament theory, which we're going to talk about next, basically creates that action of the actin sliding over the myosin and producing those structural changes in the muscle. So with the muscular system, again, we need first, in order for the muscle to contract, the first thing that has to happen is you have an action potential. Okay, so all an action potential is a discharge from the motor nerve signals that signals the release of calcium. Okay, so we need calcium for muscle contraction. Calcium gets released from the sarcoplasmic reticulum, it's that structure inside the myofibril, into the myofibril, and that causes tension to develop. And that tension is based upon the sliding filament theory of muscle contraction. So what this theory states that the actin filaments at the end of the sarcomere slide inward, just what we talked about on the previous slide, pulls the Z lines towards the center and shortens the muscle fiber. So we start with an action potential. The action potential releases calcium from the sarcoplasmic reticulum, and you end up getting this shortening contraction. And an action potential is essentially like an electrical nerve impulse. That's essentially what it is. So that kind of starts this whole process of the sliding filament theory. And you're going to have multiple phases of the sliding filament theory as you kind of go through that. So we're going to be outlining that in the upcoming slides. So here's an example of what the structures look like in a stretched muscle. So if we lengthen the muscle, okay, again, look at all your structures. So again, orientate yourself, and again, you're probably going to have to go back and look at these. So it's important to go back, look at the different structures, kind of understand where they fall and what their responsibilities are, okay? So again, here you have your A and I band, okay? So the A band contains your myosin filament, your I band has your actin filament, here's your Z line, here's your H zone. So this is we're essentially looking at a sarcomere right here, okay? This is a sarcomere. This is a contractile unit, okay? So in a stretched muscle, so if you think about this, the muscle's now elongated. So it's not shortened, it's elongated. So you could see the actin and myosin are a little, are pulled apart. They're a little bit away from the other. The I bands in the H zone are elongated. Now with this, there's low force potential due to the fact that you don't have what's called a cross bridge. So we're going to talk about this. When a muscle shortens and contracts and starts to produce force, you form what's called a cross bridge. Well, there's reduced cross bridge actin alignment because of the fact there's so much separation here and the muscle's lengthened. Okay, so this is an example of a stretched muscle, low force potential. Okay, now you notice the difference. Okay, so now you notice that this, just like we talked about, okay, look where the actin filaments went to, okay? So change in the Z line, the H zone is smaller now because the actin has slid over the myosin, okay? So this is when a muscle's contracted, okay? And here it's contracted partially. The I bands and the H zones are shortened. Now you're getting greater force potential. And the reason why you're getting the greater force potential at this point is because of the cross bridge alignment. So now what you end up have is a greater potential for force, because now you could start to get that cross bridge formation in here with the actin and myosin filament. Here's full contraction. Okay? So the fully contracted muscle. Now, when a muscle's fully contracted, you now actually have, again, reduced force potential. And the reason for that is because you're basically using up all your cross bridges. Okay? So it's really under the partial contraction where you're getting a more optimal ability to produce force. Here you're going to have low force potential. But again, what you could see is what happens when the muscle's fully shortened. So if you look at this compared to what the previous slides look like, you could see the dynamic change as far as, you know, where the actin is, the myosin, the Z lines, what that all means, and those other structures that we talked about. Okay, so here we get into kind of the stages involving the sliding filament theory. So all these anatomical structures that we discussed in discussing the myosin and actin filaments and their movement is all going to we're going to get into a little bit more detail as far as the steps. So again, this is probably going to be something you're going to have to go back and look at again, you know, between, you know, listening to this recording and also kind of just reviewing in the text, because there's some physiology involved. But you know, you'll get to know the major steps involved here. So the resting phase. So during the resting phase, there's little calcium present. All the calcium is bound in the sarcoplasmic reticulum. So there's very few myosin cross bridges bound to the actin. Okay. So actin and myosin can interact a little bit at rest, but for the most part, until that calcium gets released and there's some other actions that take place, there's pretty much weak bonding, and that's when the muscle's pretty much at rest. The excitation, contraction, coupling phase is when calcium gets released, it binds with a protein called troponin, and that causes a shift in another protein called tropomyosin, and that's when you're going to get myosin attaching to actin and the actin getting pulled towards the center. Okay, and again, the muscle force produced is related to the number of myosin cross bridges bound to actin, so that'll dictate the amount of force. But this is where you're actually getting the beginning of those filaments starting to slide over one another. Okay, so the action potential has come to the muscle, calcium has been released, and you're starting to get the coupling occurring. During the actual contraction phase, this is where the pulling action, or what they call the power stroke, comes from. Now you're going to start to use energy sources. Your energy source is going to come from what's called from your immediate energy source, your adenosine triphosphate. That's your immediate source of energy. So a good example of that would be if you go and do a vertical jump, try to jump one time as high as you can, the energy you're going to use for that's going to come from ATP. Okay? So ATP, adenosine triphosphate, is going to break down into adenosine diphosphate. Okay, and that's going to produce the power stroke for the pulling action. As long as calcium is bound to troponin, you're going to have contraction. Now, as time goes on, you may need to use other energy sources. So depending upon the length of the muscle contraction and the intensity, You may also start to include the glycolytic and oxidative energy metabolism. So those are your other energy systems. So again, ATP will supply it initially. As time goes on, depending upon the intensity and how long you hold it for, you may need other energy sources. Then you get into the recharging phase. So muscle shortening is only going to happen when that sequence of calcium binding to troponin and the coupling of the myosin cross bridge with actin and the power stroke happens. So again, and this could be repeated over and over again as long as calcium is available from the myofibril and as long as ATP is available. Once those energy sources start to break down, you're eventually going to get into the relaxation phase. But during the recharging phase, you could continue to have this process happen as long as sufficient energy is present for the muscle contraction. Then you get into the relaxation phase. So this is when the stimulation from the motor nerve stops. Calcium is basically going to get taken back into the sarcoplasmic reticulum. You're going to get resetting of the myosin head position, and that's basically going to put the muscle back into a resting state. Okay. So those are the 5 phases. Again, it's important to kind of see that. Your book talks about the steps of muscle contraction. It just talks about some of the events that occur when that we just talked about, beginning with the resting phase. So this is basically how muscle contraction will happen, starting for the different phases. Okay, so we kind of wrapped up with the last section looking at just the muscles and kind of how the muscle goes through contraction. We examined the structure of the muscle. We also looked at the skeletal system as well. So this takes us into the neuromuscular system. So now we're going to look at the interaction between the nervous system and the muscular system. And probably one of the more basic things to look at is obviously the activation of muscles. So we know that our nervous system produces muscle contraction. That's one of the jobs of the nervous system, is to help produce movement. So what you have with this, and we kind of led into this a little bit, but it starts with an action potential. Okay, so the arrival of an action potential at the nerve terminal causes the release of a neurotransmitter called acetylcholine. So acetylcholine will be in between the junctions between nerves. That acetylcholine gets released, and that produces an action. So once a sufficient amount of acetylcholine is out, the action potential gets generated across the sarcolemma, the muscle cell membrane, and you'll get the fiber to contract. Okay, and in the case of this, so this is how the control of the muscle depends on the number of muscle fibers in each motor unit are present. Okay, so in other words, in different muscles, there's a different amount of motor units. We talked about a motor unit. So if you remember what a motor unit is, the motor unit is the motor nerve and the fibers that it innervates. Okay, so that action potential comes from the motor nerve. It innervates those muscle fibers. Now, motor units work under what's called the all or none principle. Okay, so what this means is that once there is sufficient stimulation for a motor unit, okay, remember what a motor unit is. A motor unit is the motor nerve and the fiber or fibers that it innervates. In most cases, it's more than one muscle fiber. Either all of those fibers contract or none of them contract. Okay, so that's an important part of this. Now, that doesn't necessarily mean that if you're one of the confusing parts about the all or none principle for students is they hear all or none principle, they think that, well, then does that mean that an entire muscle has to contract maximally or else it doesn't contract at all? No, we're only talking about the individual motor unit. Remember, there's more than one motor unit in our muscles. Okay, so that's why we could have grades of contraction when we're looking at the muscle as a whole. Okay, because we can just activate a certain number of motor units. But the motor units themselves will either all be, individually, they either get activated completely or not at all. Now, when we look at muscles and motor units, okay, muscles that have function with great precision, okay, so if we think about muscles that would have great precision, the small muscles of the hand would be a great example, okay? those muscles could have as few as even possibly one muscle fiber per motor neuron. Okay, so most of our muscles will have more than one, but the muscles that have more precision, the reason being is that typically for those muscles, they don't have to do, they have to do some very precise work. If you think about your hands, okay, just something as simple as, you know, picking up a pencil and writing, okay, doesn't require a lot of force, but it requires precision. So the number of muscle fibers per motor unit is smaller in a muscle that functions with precision versus one that has more gross motor activity. So take like the quadriceps muscle, okay? So the quadriceps muscle, big muscle in the anterior portion of the thigh, those motor units, they may have several hundred fibers in one motor unit, okay? So the more precise the muscle has to function, the fewer number of muscle fibers in a motor unit, okay? So that's an important thing. And as we talk about next, when the motor units get stimulated and kind of the actions, it'll kind of continue to make more sense. So here we're looking at what happens when a motor unit is stimulated. So I'm going to describe what's on the slide in general, and then we'll kind of break down what we're examining here. So A, okay, so this little hill here represents a twitch contraction. B represents what we call a summation or a twitch summation. C is unfused tetanus. D is a fused tetanus. Okay, and when we're looking at this graph, we're looking at force versus frequency. So how much force and versus the frequency of stimulation as it occurs. So what we notice with this is, so what this is representing are different kind of phases or different ways that the motor unit could kind of respond based upon how much input is coming in and the frequency at which it's being applied. So every time you have an action potential coming through a motor neuron, it's going to result in a short activation of the muscle fibers, okay? A brief contraction is referred to as a twitch, okay? So a twitch is going to happen when you get the activation of the sarcolemma. As we talked about, you're going to go through the processes of the sliding filament theory, okay? Now, what's going to happen is you're going to get the same processes that we talk about. If a second twitch is elicited from the motor nerves before the first one completely relaxes, then you get a summation. Okay, that's B. So when we were looking at B, we said that's when we get summation of two twitches, and the resulting force is greater than that produced by a single twitch. Okay, so you're gradually starting to increase now the amount of force. Based upon that, you have a more frequent application here. Okay. If you continue to decrease the time between twitches, you're going to get greater and greater summation. Okay? So as the frequency increases, okay, you're going to gradually start to get, this would be an unfused tetanus. Okay? If it continues to increase more, you get a fused tetanus. And at the point of a fused tetanus, okay, that's where you will get a maximal amount of force that the motor unit can develop. So all we're saying here is the more frequent the input from the motor units, the greater the amount of force. So this is where we get into how much force you're looking at applying behind a muscle. Okay? So, and again, we kind of grade force based upon functionally what we're doing. Okay? Again, you don't put as much force behind picking up a pencil as you do, you know, in your hand, as much as you do, you know, maybe trying to open up a jar that's difficult to get open. Okay, you stimulate motor units differently, and that's what this graphic is showing you. Okay, also looking at aspects of the nervous system with their interaction with the muscles are your muscle fiber types. So motor units are composed of muscle fibers that have specific characteristics that determines what they're more suited for. For instance, we have muscle fibers that are more suited for things like force production, speed production, power production. We have muscle fibers that are more efficient when it comes to, say, endurance activities. Okay, so and that determines more than when you're just looking outside of like sports and exercise. Okay, so even just looking at like how muscles function in our body, we have, for instance, muscles that are more active with our normal posture, which would have a certain amount or have certain muscle fiber typing regardless what we did, versus muscles that are just typically more responsible for just, again, more powerful movements. And by that, I don't necessarily mean like what happens in sports. I mean just day-to-day life. We have muscle fibers that, say you have someone who's relatively sedentary and they just have to pick up a box that's heavy for them. Well, they have muscle fibers that are more inclined to do that and be more involved with that than just, say, sitting with their shoulders held in a good position for posture. Two different things. So we have three types that we're going to kind of look at. We have type 1, which is slow twitch, type 2A, which is fast twitch as well, and we have type 2X, which is also fast twitch. You will sometimes hear them called type 2B. As I'm looking at this right now, this is a typo. This should be type 2B, just like this is type 2X. They call them now type 2X. I don't know why that change happened. They used to be called type 2B. But each one of these muscle fibers have different physiological and morphological characteristics that make them distinct in what they do. Now, what I'm going to tell you is, for the purposes of this discussion, I'm not going to go through each individual characteristics. There's a table in your textbook, page 10, table 1.1. I want you to look at that and see what are the characteristics of the fiber, and look at what that would, think about what that would mean as far as what their functional capacity would be. Okay? So make sure you review that. If you're just, you're kind of just taking notes for right now, take a note to go back and look at that, or pause this and go and take a look at that and see if, you could kind of understand the different levels of each. So in looking at this, if we're looking at this in regards to exercise, and again, I think this could be important for, just about anyone taking this class. So the important thing is we understand what the recruitment patterns are. So even looking at activities of daily living, even if you're not looking at this from the standpoint of exercise, being the issue, even activities of daily living, our motor unit recruitment is going to be such that it matches whatever task we're trying to do. Okay, so I've made a couple different examples throughout this. Picking up a pen versus trying to twist the top off of a jar that's sealed really tight. Things like that. So the motor unit pattern is going to dictate how much force we produce. So, and again, force output can vary in two different ways. The force output can be varied through a change in the frequency of the activation of individual motor units, or a change in the number of activated motor units. Okay, and we talked about those two concepts before. So if that's not making sense, go back and look at what we said about how motor units operate and how they're recruited. Okay, this is kind of summarizing how that kind of functions in day-to-day life. Okay, so two ways it can vary. We can vary our force output. We could change the frequency. If you remember going back to that chart, right? As the frequency of stimulation went up, what happened to the amount of force? It went up. Okay, or we change the number of activated motor units. So say, again, going back to the two examples, say, in a certain muscle in your hand, you activate 3 motor units to pick up a pencil, and whereas you'll activate 20 motor units trying to open up a tightly sealed jar. Okay, those are completely arbitrary numbers. It's just to show a point as to how you do this. Okay, so very important. So this is the application of all that information we talked about regarding it. Okay, so the type of unit is going to be based upon the type of activity. So you could actually look at, here's something that could also help, and again, you should do this anyway, but In table 1-2, it actually goes through the involvement of muscle fiber types in sport events. So we're talking about all this with motor units and motor unit recruitment. It'll actually tell you the type, this will complement that other chart with muscle fiber types, the type of fibers involved in different sporting events. So that could kind of also give you some ideas as to the application of this through various types of activities. Okay, so now we're going to look at some specialized receptors. Proprioceptors are specialized sensory receptors that give information to the nervous system. And part from the standpoint of the musculoskeletal system, what this does, it helps to do two big things. One, it helps to maintain an optimal muscle tone for the activity we're doing and allows us to perform complex movements. So again, we want our movements to be coordinated, and part of the job of these specific sensory receptors, I'm going to talk about a couple of sensory receptors that our muscles utilize, it's to help with that movement coordination. So this particular proprioceptor, the muscle spindle. So again, if you kind of orientate yourself to the picture, so here's your muscle, okay? It's showing you the muscle spindle right here, okay? And you have two different types of fibers within muscles. You have what's called the intrafusal fiber, that's inside the muscle spindle, and then you have the extrafusal fiber, okay? When a muscle gets stretched, okay, so muscle spindles are sensitive to changes in muscle length. Okay, that's an important point with muscle spindles. They're responsible for changes in muscle length. When a muscle gets stretched, that muscle spindle deforms, it causes activation of the sensory nerve. So if you're following the picture, you see the sensory nerve, okay? This up here is representative of the spinal cord, okay? This is what we call the dorsal horn of the spinal cord. This is what we call the ventral horn. This is in the back, this is in the front, okay? So when this muscle is stretched, so we take this muscle and lengthen it, the intrafusal fiber sends information via a sensory nerve to the dorsal horn in the spinal cord. The information then gets sent out via the ventral horn through a motor neuron. Okay, so sensory nerve senses things, a motor nerve produces movement. This causes a muscle to contract. Okay, so if you had to sum up a muscle spindle and what it does, the simplest way is a muscle spindle is responsible for sensing muscle length. It responds by contracting. Okay, so if you get a quick stretch of the muscle, what the muscle is going to do is contract. Okay, a good way of kind of showing at least one of the applications to this, there's actually two good ones I think that people will appreciate, depending upon what your interests are. When you're looking from the standpoint of performance and strength and conditioning, this is what's involved with the idea behind jump training and plyometrics. You get that fast eccentric contraction, and we look to use the nervous system to produce a quick concentric contraction or a quick shortening of the muscle through plyometrics and jump training. Where you would see this just medically, a simple example of this is when you're getting your reflexes tested. Okay, so when you're sitting on the edge of the table, and you're getting a neurological exam getting done, and the healthcare professional, regardless who it is, taps the patellar tendon, and you get that little bit of a contraction, that's actually setting off the muscle spindle, okay? Because when you tap the patellar tendon, or whatever tendon, depending upon the reflex you're testing, what that actually does is it promotes a small stretch reflex, which produces a contraction. So that tells them that at that level, that reflex is present. Okay, and you look to see either an absent response or an exaggerated response, and then that would determine what the pathology would be. So again, muscle spindle responds to changes in muscle length, responds by contraction. Now, our other big proprioceptor in the musculoskeletal system is the Golgi tendon organ. Okay, so the Golgi tendon organ, so we know that it's located in the tendon. You can see it on the illustration here. So there are proprioceptors located in tendons near the myotendinous junction. Okay, again, it's a sensory. The sensory neuron is going to activate it. Now, what the Golgi tendon organ does is it actually inhibits contraction. Okay, so a Golgi tendon organ, so we kind of gave a little summary with the spindle. To kind of sum up what the Golgi tendon organ does is the Golgi tendon responds to tension and responds by inhibiting or producing relaxation of the muscle. Okay, so again, anatomically you could see where this is, and we kind of it's similar to the last picture as far as how it interacts with the spinal cord, but now we're actually getting inhibition from this. Okay, so this would be somebody who's like really straining to do something. What it actually is it's a protective mechanism. Now, sometimes it's overly protective. In other words, it shuts a muscle down probably even sooner than it has to in many instances. But it's a protective mechanism to prevent damage occurring to the tissue. You know, if you watch two individuals arm wrestle and they're stuck for a period of time, say just in the normal starting position, say they're both kind of even going at it, and then all of a sudden you see somebody kind of fatigue and go down. Well, part of what's going on there is the Golgi tendon organ is giving out, okay? And it's giving out to prevent that from happening. So again, this is more of an inhibitory response, whereas the muscle spindle is more of an excitatory response. The muscle spindle produces contraction, the Golgi tendon organ produces relaxation, okay? One of the reasons why when we're doing, you know, here's again another application to activity, is that when you do stretching for a muscle, okay, typical static stretching protocol or most stretching protocols are, you know, gradual lengthening of the muscle. The reason why we want to do that is we want to limit the excitation of the muscle. We actually want the Golgi tendon organ to override the muscle spindle, okay? So that's what we want to kind of look to do as a part of the, you know, the stretching or flexibility process. So that's kind of how those two things interact. Because, you know, what you could be saying to yourself is, well, you know, if the muscle spindle, you know, is length, when we stretch, we lengthen muscle, how come the muscle's not contracting? The key behind that is that you do a gradual lengthening so that the Golgi tendon organ senses tension and allows for relaxation to occur, kind of overrides that response. So here's a discussion question that I want you to look at. How can athletes improve force production? Again, if you're in another kind of area of study concentration and you're not concerned with athletes, just look at how anybody can improve their force production, okay? Even if it's not somebody who trains exercises, if it's a sedentary person who is rehabilitating an injury, they have to have some ability to produce force, okay? Carrying groceries requires force production. So what are the different ways? Now, again, the focus here is a little bit more on athletes because we're talking strength and conditioning. But again, it's not to imply that being strong, you know, having a good level of strength isn't important beyond being an athlete. But I want you to look at what are the ways. Your textbook talks about this, but it's a little discussion question I want you to do on your own. How can athletes improve force production? Okay, talk about that. We'll have a little discussion about that later on. All right, so that takes us to looking at some of the structure and function of the cardiovascular system. So just first of all, to start with, just the general roles of the cardiovascular system. So the cardiovascular system is responsible for, you know, transporting nutrients throughout the body, helping to remove waste, maintaining acid-base balance in the body, which is very important in both fluid and temperature regulation. So here we have a picture kind of showing you, so you know the cardiovascular system starts at the heart, and it's a muscular organ, contains a specialized muscle called cardiac muscle, and it's made of two interconnected but separate pumps. So we're going to be going through here is the kind of the way blood flows through the heart and through the body and kind of the actual track that it takes. So that's one of the things you're going to have to be able to do is know how kind of blood flows through the heart. Now, it's important that you kind of understand the order and why it does it, because I'm going to explain it one way here, but it doesn't matter where your starting point is. You should be able to go from any point and be able to describe how we go. Now, we are going to describe it starting at the right atrium. So in the heart, you have the right atrium, right ventricle, left atrium, left ventricle. So here's the way blood will kind of travel through the heart, starting at the right atrium. So blood starts in the right atrium, gets pumped into the right ventricle. From the right ventricle, it's going to go to the pulmonary artery. Now here's an interesting point. So with the cardiovascular system, when we look at the circulation in the body, so this is just an aside. We talk about arteries and veins, okay? So in general in the body, arteries carry oxygen-rich blood, veins carry deoxygenated blood. There's one exception to that rule. This is really important, okay? There's one exception to the arteries oxygenated, veins deoxygenated, and that's the pulmonary artery and veins. So the pulmonary artery takes the deoxygenated blood. Okay, again, that's the one exception to the artery rule. Okay, that deoxygenated blood goes to the lungs. Okay, at the lungs, the blood receives oxygen. So that's where the oxygen exchange takes place at the lungs. From the lungs, the blood goes to the pulmonary veins. Again, the pulmonary veins have oxygenated blood, which again is the exception to the rule in the rest of the body. From the pulmonary veins, blood goes into the left atrium. From the left atrium, it goes into the left ventricle. And from the left ventricle, that blood gets pumped out through the aorta and the circulation into the rest of the body. Blood goes out to the rest of the body, carries oxygen and nutrients, comes back, and then we start back over again at the right atrium when it's going to circulate back through. Okay, so that's kind of the order of the circulation. Again, I gave the little aside just so you could understand the difference between the arteries and veins in the rest of the body versus the pulmonary artery and veins. But you should be able to know that order. So in other words, say I start it with the left atrium, and I said take blood from the left atrium all the way through the circulation, and then ending at the left atrium. You should be able to do that. Okay, so understand that order, why it does that, and make sure you could do it coming from either direction. Now at the heart, there's also important valves. So you have the tricuspid valve and the mitral valve. Okay, so the tricuspid valve is between the right atrium and right ventricle. The mitral valve is between the left atrium and left ventricle. And again, that's just to regulate that blood flow. Okay, so as the heart beats, it's gonna basically push the blood through those valves. You also have the aortic valve and pulmonary valve. Okay, so the pulmonary valve is in the right ventricle, because that's what's going to pump blood out to take it to the lungs. And then the aortic valve is the valve that is going to going to be where the blood goes out to the rest of the body out of the left ventricle. And again, these valves open and close passively depending on the pressure gradient. So the pressure gradient, as the heart is pumping and pushing blood through, is gonna cause those valves to open up passively. Now, another point about the heart too is it has its own conduction system. So it controls its own mechanical beat. So there's an actual conduction system that we're going to talk about, that it's basically an electrical circuit that runs through the heart that regulates when things beat. So that's an important as far as the function of the heart, and that's what we're going to be looking at next. So here's the electrical conduction system within the heart. So it's got its own little conduction system that regulates the beating of the heart. So, and that's going to start. So if we look at this, you have your SA node or sinoatrial node. That's where it starts. That's where the impulse originates. So that's kind of when you hear people talking about someone gets an implanted pacemaker, well, the SA node is the natural pacemaker, if you will. Okay. So, it's when that conduction system goes bad that people have to have an artificial one put in, okay? So we're going to start at the SA node, and then that's going to take us to the AV node. So the SA node sits right on the top of the right ventricle. or right atrium, I apologize, the right atrium, so the right top of the right atrium. The AV node or atrioventricular node is between the right atrium and right ventricle, okay? So that's a little bit of an impulse delay before the ventricle. So remember, the atria contract, the ventricles contract, so there has to be a little bit of a delay, okay? Then from the AV node, or the atrioventricular node, the next area it's going to go is the AV bundle, which takes the impulses to the ventricles, okay? And then you have the left and right Purkinje fibers, which further take that out, okay? And that's going to be the system that kind of regulates the heartbeat so that we have this coordinated contraction of the heart. And there's various pathologies that you'll certainly learn about in other classes to certain degrees, depending upon what area of study you're in, that, you know, can cause issues with the conduction of the heart.